Yeah, so as Daniel mentioned, I've been analyzing the data from the macroinvertebrates that NOAA and company have been collecting for the last 3 years. So I'm going to talk about the 2022 data, but I always am talking about it in the context of the full timing, the full span of monitoring. And I don't know if you folks would have read my annual report anytime recently. It is rather large. And so I'm doing my best to kind of distill this down to what is maybe the most pertinent or the most interesting. But of course, people are from so many different agencies and, you know, probably have their own particular interests in different sets of streams. So if there's anything that you see, you know, in the report or something that you have questions about, you know, you're super interested in Abernethy, or for example, and I'm, you know, maybe I don't talk about that much, feel free to reach out to me, and I'll be happy to provide more details or answer any questions that you might have. And then I can't really see, Daniel, you can kind of decide, you know, I just sort of have everybody shoved off in a bar at the side. So if we want to handle questions as they come up or wait until the end, I'm kind of good either way. You might make the call on what would be the most efficient way to proceed.
I think it might be better just to wait until the end, let you get through your presentation, and then everyone write down your questions.
Okay, sounds good. So just to give you a little bit of a refresher, nothing has really changed about the methodology, but I'll just kind of run through quickly what the methodology is, because it's a little bit different than potentially the sort of standard stream monitoring that you might be more familiar with. Give you an overview of what just that 2022 macroinvertebrate community looked like. Talk a bit about the sites, what looked good, what looked moderate, what looked not so good. And then I want to spend you know, probably the majority of the time, now that we have projects that have been implemented and we're anywhere from 1 to 3 years post-implementation. Of course, this whole thing is about effectiveness monitoring, kind of home in on some of those and take a look at the pre and post project communities. Did we see any changes in the communities or any of the community metrics? And if so, what do they tell us? Do they tell us anything? And then just sort of summarize the trends from this year's data and the full 3 years of data at the end. So standard sampling span, we're kind of in the middle of it right now. So the streams were sampled from July through September, prioritizing sites that were known to dry down early. And in every year, for a site that was sampled in any previous year, NOAA tries to sample it within the same 1 to 2 week period, which has generally been possible, just so that we don't have temporal variation, introducing more noise into the data set. And last year there were 41 sites, that's 24 project and 17 control. Because you can all do math, you're going to notice that that is not the same number of project and control sites. A couple of sites where the project implementation is pretty far in the future, potentially not even within the 6-year span of this particular project. Details on the project are a little fuzzy, and so controls have not necessarily yet been selected. And there are a couple of project sites that share control sites that are spatially co-located or close to being co-located. So that's why you don't see 24 of each. And then always to make sure that we have a robust sampling technique, that there is not, you know, operator error, or that there aren't kind of vulnerabilities in the protocol where maybe it's implemented differently from year to year. Duplicate samples were taken at 4 sites for quality assurance, and that represents about 10% of the total number of sites sampled, and that's standard for what the Oregon Department of Environmental Quality suggests. So of those 41 sites that were sampled, it was the 2nd year of sampling for about half of them, and the 3rd year of sampling for about half of them. And within the period between the 2021 and 2022 samples being taken, new projects were implemented at 3 sites. So all of the projects are fairly young, but we do have projects that are of differing ages. So this is the way the stream reach looks for sampling. We've been using a 30-meter reach, so it is fairly short, but it also kind of represents where the projects are being implemented and what their footprint might be expected to be. And because we have such a variety of streams here, a lot of them are low gradient, some of them are tidally influenced. In some cases, the project has involved reconnecting a side channel. So we have a variety of flows. And that means that the standard riffle-targeted habitat was not deemed to be representative, really, of the goals of this project. We're not really looking necessarily at biotic integrity, which is one of the reasons, or biological health, which is one of the reasons that riffle samples are taken. We're looking to see changes following restoration, and not all that restoration is going to affect flow or affect riffle quantity. And some of the reaches simply don't really have riffles. So this is a reach-wide multi-habitat sampling. So we're hitting whatever array of habitats and substrates are there. So the reach length is set, transects are set at regular intervals along that reach, and then one 1 foot squared net set, one dip net sample is taken in each one of those transects, working, of course, from downstream to upstream, and alternating between the left, the right, and the center of the channel, so that you're also getting some random kind of spatial movement. You're not just sampling, for example, down the Thalweg. And then at each point where a sample is taken, the flow type, the substrate composition, and the water depth at each of those points is recorded. And of course, these 8 net sets can contain quite a few macroinvertebrates. And so the standard protocol is that they are randomly sub-sampled in the wet lab down to a total count of 500 organisms. And that's what is identified to species or genus or to family. And then that's what I get back. So the overall restoration inventory at this point is that restoration has been done at a total of 7 sites, and that's been done kind of prior to a few of the different sampling periods. So we had one site where the restoration was done before the very, oh, sorry, Roy. Oh, not a question, just a noise. We had one site where the sampling was, the project was implemented prior to the very first set of samples being taken. Another, 1, 2, 3 that were implemented between the 2020 and 2021 sampling period, and then 3 more that were done between the 21 and 22 sampling period. Hopefully that makes sense. And there was a lot of variety in the type of restoration activities that went in, things that might be considered to have rather more impact on substrate and flow and biota, like dam removal and side channel reconnection, and others that might be a little bit more localized in nature, such as artificial riffle creation or placing large woody material. And we'll talk about those projects in detail a little bit later in the presentation. So when doing the community analysis, of course, we can't apply those models, the IBIs and the predator O/E scores that were developed specifically for riffle targeted systems. And again, that doesn't necessarily get us to it doesn't really get us the answers necessarily to the questions we're asking about restoration here. So I use a combination of both taxonomy and trait-based or ecological community measures, I guess you could say, or factors. So, the taxonomy stuff is kind of standard: community evenness, richness, number of species of different taxa, similarity indices, things like that. And then looking more at kind of what gets a little bit into the responses to habitat, or tells you a bit more about the habitat: flow and temperature associations, sensitive and tolerant organisms, the type of feeding guild that they use, how they move, and what their life span is. So volatinism, is it 1 year, less than a year, more than 1 year to go from egg to adult? And those all tell us different things about the habitat and about the way the community is changing. And this is a figure from the paper, and it's my obligatory figure that I have to apologize for because it is super duper busy, but this is a cluster dendrogram of the macroinvertebrate community at all of the 41 sites in 2022. And anything that is in black text is a control, and anything that is in teal or aqua text is a project. And this is just kind of a visual reminder to me to say that when we look at just the data from 2022, those communities in all but 2 of the designated project control pairs were most similar to each other. In other words, they formed, a single 2-pronged fork on this dendrogram. So that's kind of a good indicator that, even though you can see we've got different clustering, more or less related samples here that kind of group themselves out, it's a bit reassuring that our project and control pairs actually are similar to each other, kind of reinforcing that good good designations were made. And then back to that kind of quality control situation, all of the replicate samples and those duplicate samples are taken in the same reach at the same time as what you might call the initial sample. So they're taken at the same time. All of those communities were most similar to each other. So again, reinforcing the fact that we have a good, robust sampling technique, reassuring us that differences that we see in the community are more likely to be due to real habitat or stream differences and less likely to be due simply to the way in which the samples were taken. And then this is a principal components analysis, just kind of showing you the way some of these different sites cluster out. Again, the teal dots are project sites and the black dots are control sites. And so you can see, for example, if you look at the upper right quadrant, right, and you can see that that's where like Eagle Creek, Metro Clear Creek is, that the long axis there says Rithregina, and that's a flat-headed mayfly, more associated with colder, faster flowing waters. So you can kind of get a little bit of an idea of like, oh, you know, that's what the habitat is a little bit more like in what are admittedly a few of the higher quality streams in this data set. If you look off to the upper left-hand side with a big cluster of project sites, you can see that those longest vectors belong to non-biting midges, very tolerant Chironomini and to Annelida, which are segmented worms. So you can get an idea that there's maybe some difference in the ecological quality of these different clusters of sites based on what the more dominant types of taxa were. So just kind of another way of seeing that separation between the sites. So overall, we had a total of 204 unique taxa that were taken in 2022, which is a rather more than 2021. And that doesn't mean that any more sites were sampled. There was actually a very similar number of sites sampled. I think it might have been, it's like a one site difference. It was either 40 or 42, I think, last year, or in 2021, and then it was 41 in 2022. As in past years, the greatest diversity that we're seeing, the greatest taxonomic diversity, is among the Diptera, the true flies, as well as the Trichoptera, the caddis flies. And there's a big range in total sample richness, from as low as having only a dozen unique taxa, genera or species in the entire sample, all the way up to 47. And just to kind of break that down a little bit further, if we were to use the invertebrate race index of biotic integrity that DEQ has developed for riffle targeted samples, one of the metrics in that is sample richness, and the highest scoring streams in that metric, in that model, they would score the highest for sample richness if they had 36 or more taxa. And so about half, a little over half of all the samples that were taken did have that higher number of taxa. Sites that have fewer than 20 taxa receive the lowest scaled score in that IBI model. And so there were two samples that had that low number. I think they both had 12, or it might have been 12 at Asadero and 14 at Renierson, but pretty low richness there, pretty low numbers of taxa, but a large range among all of these sites. Non-insect organisms, so that's things like worms, snails, pea clams, isopods, scuds, things of that nature. If you have a high abundance of those, it tends to indicate a more degraded system. So that's just another one of the factors that I kind of look at overall. So non-insect organisms did comprise more than 50% of the total organismal abundance at about a quarter of the sites. More of those were project than control, 8 project and 3 control. And that's actually a little bit less than the number in 2021 that had that much in the way of non-insect organisms. Overall, not terribly surprisingly for highly urbanized streams, the diversity of mayflies, stoneflies, and caddisflies, the EPT was fairly low overall. There were 2 sites that had absolutely none whatsoever of any of those groups, kind of similar to what we saw in 2021. Stoneflies, of that big 3, stoneflies do tend to be the most sensitive, the first ones to drop out of the system. There were 17 sites out of the 41 that had no stoneflies at all, but that's actually a little bit fewer than the number that lacked stoneflies in 2021. So maybe a little tiny bit of a positive indicator there. As far as the functional feeding groups, the most common were the collectors. That's collector filterers and collector gatherers that feed on that organic material, that suspended organic material. Shredders were by far in the minority. And so those are the critters that would feed on the leaf material, plant material that falls into the stream. So that can be an overall indication that there's not a super high level of riparian inputs. And most sites were dominated by pretty cosmopolitan taxa, fairly tolerant taxa that can survive in a broad range of conditions. And looking at that, you can see that a lot of those represent those non-insect organisms that I was talking about in the previous slide, like the pea clams and the snails and the worms. But the dominant organisms, so the one that dominated the numerical abundance at 12 different sites with kind of an even split there with project and control, was actually a caddisfly, a mayfly, or a stonefly. Although I actually don't think it was a stonefly. I think that was mayflies and caddisflies. I don't believe we have any stonefly dominated sites as of yet, but it gives you something to hope for. Kind of taking a look at, are the project sites better or worse quality than all the control sites, kind of considered as two different pools. The project site communities, so this is just the overall taxonomic community, they differed more from each other than all the different control sample communities did. Although you can see there's still a pretty high overall average dissimilarity among all the project sample communities and all the control sample communities. But the community composition was much greater and significantly different between all the project versus all of the control sites. And all of those average dissimilarities among all those different groupings was higher in 2022, suggesting that there might be greater community variation, which might be due to climate, temperature and flow and precipitation. It might have factors from restoration thrown in there, but there definitely are some changes going on. And then of all the different community metrics that I look at for these groups, the means of just 4 of them were significantly different, again, between all control versus all project sites. And they did reflect better overall conditions in the control. So you can see the minimum and maximum values for each of those 4 metrics, 3 of which are fairly related: the number of caddisflies, the EPT, and then the percent of all taxa in the sample that were comprised of mayflies, caddisflies, or stoneflies, and then that relative abundance of non-insect taxa. So more caddisflies, more EPT, and greater relative diversity of EPT among all the controls, and greater abundance, relative abundance of non-insect organisms in the project sites. So for sites that had been sampled across 3 years, since we couldn't do this with sites that were only sampled for 2, I looked at each of the different community metrics or measures that I calculate for each individual site over those 3 years to see if there were any statistically significant unidirectional trends. So that was 22 sites that had been sampled for that 3-year period, and 39 individual community metrics. So that is a total of 858 possible correlations. 17 of those 39 metrics did not show any statistically significant trends at any of those 22 sites that had been sampled for 3 years. And 7 of those sites had no significant trends at all for any of the metrics that were calculated. So out of that 858 possible correlations, 36 significant unidirectional trends were found. It's about 4% of the total possible correlations. And looking at just across those 36, the metrics that had the greatest number of significant trends across all sites, again, come back to that percent diversity of non-insect taxa. And this is funny, I'm having a hard time. There we go. And the relative diversity of EPT. And it was a little bit of a mixed bag. So looking at the direction and the nature of these trends among the different individual sites where there were statistically significant differences, those trends suggested improving conditions at some of the sites, declining conditions at some of the sites, and kind of a mixed bag at some of the sites, with almost an even distribution among the number of different sites there and whether they were project or control. So not all necessarily trending in the same direction. So I want to kind of giving you that sort of overall kind of idea of what the community is like and how the project and control sites just in general differ from each other. I want to focus in on what we're seeing or not seeing for community changes at sites where restoration activities have been implemented. So this is just showing that list again of the 7 sites and just reiterating when the restoration was done. And so where it says pre-2020 or pre-2022, that means prior to the macroinvertebrate sampling period of that year. So restoration at Eagle Creek, that large wood placement was done before the 2020 sample was taken, and the dam at Kelly at Obrus was removed before the 2021 sample was taken. So that kind of gives you an idea of how much pre and post restoration data we have to be able to compare. And then you can see again that variety of different types of restoration projects that were actually done. So I'm going to run through all of these individually. So Eagle Creek, before the first samples were ever taken, had large wood placement. Now, this is a site where both the project and the control reaches are somewhat less impaired overall. They're a little bit higher biological quality from the macroinvertebrate standpoint, just within the entire FIP sampling set, so to speak. But taking a look at just at the communities, we don't really have baseline data from this because it wasn't possible to get in before the project was done. Just looking at the project versus community control community composition, there was no significant difference in some of those kind of more obvious metrics, such as total number of taxa in the sample, the number of those EPT in the sample, or the community temperature or community sediment optima. So you can see, if you look on the left, you can see the control and project are shown from left to right in 2021, and then in 2022, again, control and project. The blue bar is the number of taxa in the sample, and you can see those numbers are pretty high. Remember, 36 or higher scores in the topmost range of that biotic index. And they're not really different between the control and the project in the different years. And the same thing with the number of EPT. And there was no difference between those in project versus community. And then, the dendrogram, you can see from the similarity, the percent similarity there on the y-axis, so to speak, that overall the communities have a pretty high degree of community similarity. And the project and control sample from 2022 were more similar to each other than they were to either from the prior year. But we're still looking at an overall greater than 60% community similarity. So not huge differences between the communities there. With Kelly Creek at Obrus, this was a small dam that was removed. There was a year of sampling done prior to this in 2020. So we've got a couple of years now of post-restoration sampling. And so one of the things that you might kind of expect to happen when a dam is removed is that there's a difference in the flow types. It's not one big, deep sedimented pool anymore. So one of the things that I do in the report, because NOAA records what the flow type was, where each net set was taken, is that I can kind of take a look at in each year. Was it all riffle? Was it all pool? Did it change over time? And so just looking at that graph on the lower left, from left to right, prior to the dam being taken out, every net set was taken in a pool. And then there was actually a little riffle in 2021 when the dam was taken out. And then there were a variety of flows from riffle to glide to pool in 2022. So a little evidence there that the flow is in fact changed. And then if you take a look at the graph on the right, showing the total number of taxa in the sample and the number of EPT, the total number of taxa has increased with every year since the dam was removed. And we went from having 0 EPT in 2020 to 4 total taxa of EPT, or rather ET, because there aren't any stoneflies there, in 2022. So that's still a pretty low number, but it's a big increase over what we had before. And there were significant unidirectional trends for a greater number of different metrics, and most of them are trending in a direction that suggests improved habitat conditions. So increasing numbers of trichoptera, of caddisflies over time. Obviously, that includes more sensitive organisms. The relative abundance of the most common, the most numerically abundant taxon decreased over time. That is usually a sign of a more stable community, one that has rather more habitat niches. It's not just dominated, like at 80% by worms or something. So that's a positive indicator. The proportion, the relative abundance of sediment-tolerant organisms went down, again, suggests decreasing sediment. Increasing numbers of sprawlers, so insects or invertebrates that just kind of lay there on the surface. They can be smothered out with increasing sedimentation. So if their numbers are increasing, that can be an indicator that sedimentation is improving. And then the Simpson Diversity Index, which measures both the number of different taxa and the abundance at which they occur, was also increasing. So those are all in a direction that suggests a better conditioned habitat and a better conditioned community. And if you look at the dendrogram, you can see that community in 2020, immediately after the dam was removed, differs more from the communities in the following 2 years as these new flows and new habitats were being revealed. Same token, the community temperature optima went down across those 3 years, and the community sediment optima decreased as well. Although, especially the sediment optima, they are still, a little bit higher than you would like to see, but definitely some movement on the pendulum here. The Lower Johnson Creek sites. This was a large wood placement that was done between 2020 and 2021 sampling. And you can see the dendrogram down there showing that there is a bit of a split between the 2022 communities and the earlier years, with project and control still being similar to each other. But this did not translate into a significant difference in community composition between project and controls across all those years. And at the project site, there was no significant difference in community measures across time. So there were no significant trends. Interestingly enough, there were a few at the control site. The community biotic index decreased, which is generally good. That suggests that there is less organic pollution. The number of sprawlers decreasing, though, this is one that's a little bit of a mixed bag. That might indicate that sedimentation is a bit of an issue. The increasing number of predators is more of a positive sign. It means that there's a stable prey base for those organisms to be able to exploit and to survive on. So this was a little bit of a mixed bag, and the changes in the control community, but not as of yet in the project community. Very similar, some of those community measures, total taxa richness, how many EPT taxa there were, community sediment and temperature optima. They were pretty similar in project versus control within an individual sampling year and really between the different sampling years. And even though there was some change, if you look at like the temperature and the sediment optima, there was no significant difference in any of those means. Asadero North. Prior to the 2020 sampling being done. So again, this is one where we were not able to get baseline data. There were some small engineered riffles that were placed. If you take a look at the dendrogram there on the right, you can see that the project community in 2022 is now more similar to the control samples. The 2020 and 2021 projects there in teal are a little bit more of an outlier with lower similarity to the later years and to the control. But again, there was no significant difference in the composition of project versus control communities. And the total number of taxa in the sample was still greater in the control than it was in the project site. And so again, you can see starting at the left on that total taxa and EPT graph, North Fork Deep Creek was not sampled until 2021, so that's why there's not a control for 2020. But you can see that there are more total taxa in the control than there are in Asadero, and the numbers of EPT do tend to be higher as well, but they were not significantly higher. But there were some significant unidirectional increases in a few of the different metrics. And even for the ones that weren't necessarily significant, there are several of the metric values in 2022 that do suggest some improvement, just kind of looking at changes across time. So more total in EPT taxa at the project site, more clingers and organisms associated with erosional or faster flows, and a higher Simpson diversity index. At Kingfisher, there was an old side channel that was reconnected and restored prior to the 2022 samples being taken. And again, you can see there on the dendrogram that the Kingfisher project samples taken prior to restoration 2020 and 2021 differ more from all the controls as well as from the post-restoration project sample. But so far, looking just at the total project versus control community across all years, no significant difference in community composition. And there were no significant unidirectional trends in any of the metrics at either the control or the project sites. But there was an increase in sample diversity and in EPT richness in the project post-restoration. So you can see that in 2020, there were like one EPT at Kingfisher. 2021, it might have been 2 in 2020 and 1 in 2021. And then there were actually 20 members of the EPT in 2022. So that's a fairly dramatic increase. So the number of EPT taxa, as well as their representation among the total taxa, their relative diversity, definitely did increase. And once we're able to get more post-restoration years of data, I can be looking at pre and post-restoration communities, but with only 1 year post-restoration, that's not quite as possible to do. And more than half of all the taxa in that 2022 sample were EPT. So that's a big change. Again, no significant differences between the means of project versus control for the community temperature or community sediment optima, but the values for both of those community measures in the project in this first year post-restoration were actually the lowest of any sampling year, as you can see from the graph. Not terribly lower for the community temperature optima, but rather more dramatically lower for the sediment optima. Abernethy below Newell was another large wood placement prior to the 2022 sampling period. And one of the things that you can see from looking at the habitats where the flows, the flows where the net sets were taken, is that there are some areas of faster flow now. So if you look at the 3 right-handmost bars for the project site, that's 2020, 21, and 22. The amount of run and glide habitat has increased in the last 2 years, whereas in 2020, most of the samples were taken in pools or in glides. So a little bit of a change in flow there. No significant difference in community composition for project versus control in all years. And you can see that the control samples there in aqua are still, the communities are still rather more similar to each other for the projects than they are to the control samples. Significant unidirectional trends in metrics over time included actually lower total taxa, fewer total taxa in the project, a decrease. But within that, there were more stoneflies and there were more EPT. So that's a positive indicator. And in the control, there was a trend towards increasing numbers of climbers, which could factor into changes in substrate, possibly changes in vegetation in the habitat. As far as those other kind of 4 that I've been looking at, total taxa, EPT, and community sediment and temperature optima, at this point, there is no significant difference in the project versus control for the means of those. Seabed Middle, this is another restored side channel that was done before last year's sampling. And you'll notice that all of these are teal because the reach that sort of has been designated as the control, as the most appropriate control, was thought initially that was also going to be impacted by the restoration that was done. But you can see that in 2022, the post-restoration project community is more similar to the controls. And actually, you can't see that because there is an error on that graph and they all look like they're projects. So trust me, the 2022 and 21 on the right there should actually be control. So that post-restoration community was more similar to the controls, but again, there's no significant difference in project versus control composition. Significant unidirectional trends across time at that restored control. Fewer predators, which, you know, that could be possibly, you know, due to the absence of a stable prey base. This is a somewhat more impactful type of restoration, and oftentimes it can take a year or two for the habitat and for the community to adjust. So time will tell on that one, but also decreasing numbers of warm water associated organisms, which makes pretty good sense if you've got increased flow in the channel. And then post-restoration, the number of total taxa was higher, as was the number on the relative diversity of EPT. Simpson Diversity Index was higher, but also there were more worm associated and more burrowers, and that increased number of burrowers oftentimes will reflect increasing sedimentation. So this is a little bit of a mixed bag here as far as habitat indicators. And then decrease again in the community sediment optima. So that's a rather more positive indicator. So I know this is a lot of different sites and a lot of different things kind of tossed at you. I really just tried to kind of pull out and focus on the project sites where the projects had been implemented, also for the sake of time. So overall, just kind of summarize this. From year to year to year, even if restoration has been implemented, the similarity in the macroinvertebrate community is fairly high. You know, we're talking anywhere from 45, 50 to 65, 75% overall community similarity. So they remain fairly well related. So that suggests that community composition is remaining fairly stable, even though we have seen across these last 3 years some kind of wacky swings in temperature and precipitation patterns. And one thing that we always have to keep in mind with, you know, highly urbanized streams, the way most of these are, is that these communities are already experiencing a lot of strong environmental filtering. Some of which not a lot can be done about. You know, if a stream is running right next to a highway, that stream is going to be right next to the highway. There's a limit to how much remediation or changes can be made to surrounding land use and anthropogenic structures. So we may already be looking at some of these streams at communities that are already constricted or restricted, I guess, and that may give a little bit less annual variation in community composition than we sort of expect to see in macroinvertebrate communities. Communities in most of those project control pairs are most similar to each other within each sampling year, again, suggesting appropriate control designations. Some of the differences that we do see looking at project versus control sites suggest that overall the control reaches do have better habitat, do have better biotic conditions. And I should say, I talked about the significant unidirectional trends in the community metrics at the project and control sites where the projects have been implemented. I did that for all the sites, not just the ones where the projects had been done. And so if you look at the report, you will see that even at sites where no restoration has been done, or at controls, you know, for example, that haven't been altered, there are still some significant changes, significant trends in some of those metrics. So, you know, even without active restoration, there are still some changes in the community that are occurring. Of the 6 project sites that had more than 2 years of sampling available post-restoration, 2 of them did not have significant trends in any of the metrics, but 4 of them had trends that overall indicated improved conditions or improved habitat, and you can see those listed there. And not surprisingly, the greatest changes in the macroinvertebrate communities were seen at the sites where the restoration project itself had a more extensive impact within the reach or a more dramatic impact on the habitat. So dam removal, of course, is going to massively change the flows. It's going to change what the sedimentation looks like. Side channel reconstruction, right? You've just added an entire new channel that can be colonized, and that is potentially going to be rather dynamic for a period of years. But I also, want to point out that I can tell you, based on the trends that I see, or significant differences pre and post restoration, if it seems to indicate that habitat changes are for the better or habitat changes are for the worse. But Ken has heard me sing this song already this morning. Without really detailed geomorphological habitat data being collected, I simply have to infer, here's what the community looks like post-restoration. Here's the kind of restoration that was done. Here's what I would hypothesize the impact would be on the macroinvertebrate community. But without accompanying detailed annual habitat data for those reaches, you can't unequivocally relate changes in the community or changes in community metrics directly to those restoration actions. So I think I've kept within a fairly decent frame of time here so that there is time available for questions, because I know that you also have other business that you need to attend to. So I will open it up for any questions or comments.
Great, thank you, Zee. And I see we already have a question. I'm wondering at this point, maybe I should stop the recording, or should we keep the questions in the recording? What do folks think?
It might be informative.
Okay, I guess we'll start with Ken.
Yeah, thanks, Daniel. I think Zee, and I guess I had one question that I was thinking about as you're presenting, and one based on what you just said. And are what you guys call control sites, remind me, are those what we might think of as impacted, so you
Yeah,
pick them
and
because...
we've gone back and forth on whether to call them control or reference. So we selected sites, and Daniel and Noah, feel free to jump in here too, that were similar in setting, that were spatially close. In some cases, they are a different reach of the same stream, and others they are a different creek, but one that is what is nearby. At which no active, or passive restoration actions were anticipated to be implemented within the 6-year period of the project.
Yeah, thanks, because it seemed like several, the conditions were better, if you will, in the control reaches. And I guess as we compare, are you just looking at how much both are changing to see if there's a greater magnitude of change in the project compared to control, or are you hoping to see that the project site exceeds the control conditions or the quality based on the macroinvertebrate?
That is a really excellent question. Realistically speaking, for most of these streams, considering their settings and just the ongoing pressures from surrounding land use, taking a look first of all at just, is the community changing in a significant way pre and post restoration? Of course, we're still kind of early days for that, just as far as how many years of data we have. And then within that, is it becoming more similar to the control? Because as you say, in most cases, the controls do appear to be, I always hate to say higher quality, healthier, I guess,
Yeah.
Higher values for a lot of these positive metrics. As far as whether they would exceed the condition of the controls, that would be great. I don't know if that's a tremendously realistic expectation, just given all of the environmental filters that are operating at the watershed level.
Right.
I mean, some we'll be able to see for sure as we're doing the analysis, but it's not necessarily what I anticipate seeing, I will say.
Yeah. Yeah, thanks. And my last question, based on kind of what you sort of end with, have you guys been talking, maybe you don't know Zee, but you know, Jim's here and Daniel, and I'm just curious, is over time there a plan, or do you guys see an opportunity to look at the habitat data that ODF&W is collecting at these same sites to help interpret the macroinvertebrate results, and vice versa? Thank you.
Yeah, thanks, Ken. That was a great question, and it was something I was going to mention at this point to Zee, looking at, you know, maybe before we get to the analysis phase for the current 2023 collected data, Zee, we could talk about, you know, what metrics, what information might be needed to maybe help with that kind of analysis. Jim and the ODFW survey crews are also taking pre and post project data, but they are not doing it with the same frequency because, as you know, that's a much more involved set of monitoring than macroinvertebrate collection on a kind of site per site basis. So we will have pre and post project data for all the sites. It's just they won't all be from the same year. Post project for the same year.
Well, and I remember that last thing. I don't want to get in the way of other people asking questions or making comments, but there could be opportunity to do that, like the
and I think you may be frozen.
I'm never sure if that's me or the other person. But yeah, Daniel, if that, I mean, whatever habitat data is available over time, that would definitely be a good
funding
thing.
allows, do some of that analysis. You know what I mean? So that there's like the most amount of data available at that time.
Okay. Great. Let's see. Anybody else with any questions?
Dan, I'll just follow up real quick since there's no hands up. Yeah, we do the pre and post 1 year high and low flow surveys, and then at the very, say, the 7th year or the 5th, we're going to be going back in and doing another round of post surveys on a number of sites. We think we're going to hit around 6 or 7. That's to be. I had to go back and look at our commitment on our last supplemental grant agreement, but we will have pre and post 1 year, and then anywhere between, I think, 3 and 6 years on a number of sites. I think there's a general overall interest. We're just would have to figure out, you know, how we would work that through the system and create the analysis and basically do the work. But we're looking at probably, you know, year 7 or year 8 to do that. But I think there is a general overall interest.
Great, thanks, Jim. Any other questions? I see, Tom, you have your hand up. Tom, I'm not hearing you. I don't know if others can hear Tom or not. Nope. Well, Tom, while you're waiting, I'm just going to ask Zee a quick question. When you showed that dendro chart at the beginning, you know, looking at the similarities, I noticed that the Landslide Toe project was the very last one and seemed to be most different. And I'm wondering if I'm reading more into it that's there, but do you have any thoughts on that?
Well, yeah, I mean, it is definitely an outlier. I'm just, one sec. And that was actually one of the 2 sites of the entire data set in 2022 where the project and the control were not most similar to each other. But I know that is kind of a different site as far as setting and flow, is it not, Noah?
Yeah, so Landslide Toe is currently, it's an old, well, actually it was the old, I guess, main stem of the Clackamas River at one point that was filled in one of the many large flood events over the years. And so what's left there currently is basically an isolated, essentially wetland type habitat. That the project is going to be designed to kind of reconnect that as a side channel to the Clackamas as it is currently configured. And then the control site for that is I believe, a previously constructed side channel that's close to where Eagle Creek comes into the Clackamas River. So it kind of stands to reason that those would A, be radically different presently, and B, that the control would look far superior in terms of habitat quality because it's a connected side channel with flow as opposed to an isolated stagnant wetland kind of habitat.
And from your notes on the data sheet for 2022, you did note that the reach was wider than last year, it was warmer than last year, and it smelled like rotten eggs.
Yeah, it was somehow it was quite different this past year than it was the year before, and I'm not sure what accounts for that.
That channel is pretty rapidly widening, and part of the island upstream is disappearing, and the bank has eroded like, I don't know, 10 or 15 feet this year from that side. So it's pretty active right in there right now. I think that's a natural channel that's been around for quite a while. And it's what Eagle Creek actually sort of flows into. And so the upper end of it comes in just below the alcove.
You're talking the control reach.
Yeah, the control site. Yeah, yep. And I think that is natural. It's interesting. There's some natural analog wood jams in there that I've taken pictures of that look an awful lot like some of the ones that we create. So we're in the same ballpark. But yeah, it's definitely widened. I think it's starting to take more of the river, and the island upstream is getting a lot more vegetated and kind of causing the river to split more. So.
Right. Well, it sounds like this is one of those cases Zee talked about where if you had more geomorphology information, you could start to help make some interpretation.
Yeah, and it was also the only site, I believe, if I'm remembering correctly, where the substrate, where the net sets were all taken. So like Noah said, it was in still water, but it was all plants and woody debris. So that's going to be another thing contributing to a big community difference with the other sites, with that just that wetlandy nature of it.
All right. Tom, do you want to try again? And Ken had a suggestion that you could type something into the chat.
I'm going to try this. Can you hear me?
Yes.
Excellent. AirPods, they jump in and out. Yeah, so Zee, thanks. It is typed in the chat, but I'll try to be a little more precise. So Newell Creek entered Abernathy main stem, and restoration happened both within Newell Creek and within main stem Abernathy. You commented on kind of the faster flows and the habitat change, and so I think absolutely more geomorphic data would be helpful to try and understand what's happening there. But I guess my question is, would we expect as pools begin to form, I would think that pools are going to be in form of that tree and condition or something else. And I can talk more about the treatment. You probably know about the treatments. They're different in Newell Creek Canyon from what was done in main stem Abernathy.
So, the pools definitely support a different subset of the macroinvertebrate community than, say a riffle or a run for sure. But the nature of the community might change if you go from these higher flows to now having whole, increased numbers of pool units. But I will say too that one of the things that I have noticed, especially with this multi-habitat sampling, is that you might not get, if you're looking at the pools, those would tend to be organisms that by their nature are more tolerant of sedimentation, right? Or there might be more tolerant of slightly warmer temperatures because the water's just sitting there. But within a kind of a bigger picture where you've got, well, we did this restoration and now we've got higher flows, but now we also have pools, and the pools have been created by wood, and we got wood that's in there, that you actually do still oftentimes see an increase in overall diversity. So, you're concerned about, well, damn, we put in, we did this, and then there's more pools, and now it's going to look like we have, a diminished taxa because they're hanging out in the pools. If this is also accompanied by, ongoing changes in flow, by changes in temperature, by other changes in the substrate, you're not just necessarily thinking, oh, wow, pool formation, wow, that might be good for the fish, but boy, it's going to make our macroinvertebrate data look bad.
Yeah, well, I think really the tale of Abernathy mainstem and the impact of the North Knoll Canyon restoration will be told after a couple more winters, when there's been enough flow through that system to really start to shape that substrate a little more.
Sure.
This winter we were seeing a lot of flow underneath those the large woody debris that had been placed along the scallops on mainstem Abernathy, and I haven't yet revisited the canyon, so I'm kind of interested to see what happened in there. But it was not a dramatically high flow winter. I'd like to see a little bit more of that.
Yeah.
And
that's a really good point, because a lot of these systems are really dynamic, right? For the first few years after restoration, while they're kind of adjusting themselves and settling in. And so that's one of the great things about the span of this project. You might see a site that is really dominated post-restoration in 1 year by things like black flies or small minnow mayflies. It's like, well, yeah, they like disturbance, they come in first, and then you're going to see these kind of shifts and adjustments. And so that's just kind of another way of saying that, yeah, you may not see like, oh, here's this, wonderful, the heavenly choir is singing, it's post-restoration, marvelous community time. But those shifts over time are still going to be telling you more about, okay, well, we can see what's happening with measuring the flows. We can see what's happening with measuring the temperatures. What can we infer now about other things about the habitat under the water based on how that community is changing? So it is, it can still be very informative.
Sure.
Thank
you, Zee.
One minor note from the field on that site specifically as well is there were a couple of remnant beaver dams that were in place on mainstem Abernathy when we sampled in 2020 and 2021 that were gone completely last summer, whether due to just having been abandoned and finally blowing out or what have you. So that may account as well for some of the changes in the flow regime through there.
Thanks, Noah. That's a great point. I do remember those well. And I think one of our restoration sites was actually quite close to one of those locations. So we'll see if the beaver moved back in.
Great. Well, thank you. Any final questions before we officially end the presentation / Q&A session?
Well, you got the presentation, you've got the reports. So if any questions do occur to you in the interim, or as I said, if you're really, really fascinated by Metro Clear Creek, and I didn't talk about that, feel free to take a look at the report and then shoot any questions that you might have my way.
Great. Well, thank you very much, Zee. This is great. I know we all really enjoy hearing from you every year, talking about the results and helping us to interpret the results.
Glad to do it. Glad to do it. All right.